The next speaker is talking about leveraging gene editing, technologies for precision and efficient in-cell and gene therapy development. And it's Kristy Wood, who's the SVP of Technical Ops in Intellia. Thank you.
Thanks. Well, good morning, I guess the last talk of the morning. I'm happy to be here, and today's talk is really going to focus on how we have used CRISPR-Cas9 gene editing to develop in vivo therapeutics for multiple programs and indications. Quick legal disclaimer. Hopefully you guys will memorize that. There's a quiz at the end. So CRISPR-based gene editing technologies are a promising new therapeutic modality. It's a very compelling approach in the fact that we can treat patients at the root cause of their disease rather than just managing symptoms. The potential benefit of this is that we may have a single dose treatment that has that lifelong benefit for the patient, which overall can reduce burden on the patient themselves as well as the overall healthcare system. So we're excited about the progress that we've been able to make in terms of developing gene editing therapeutics. So to date, we have several programs that are in clinical development. We have 3 active phase 3 studies. Our first study is for the treatment of hereditary angioedema. This is with Intellia 2002 or Lonvo-Z. And then we have 2 additional phase 3 studies for the treatment of transthyretin amyloidosis, either the form of the disease being cardiomyopathy or polyneuropathy, and these are actively recruiting phase 3 trials. And that's with treatment with Intellia 2001 or Next-Z, and I'll show a little bit of a highlight of some of the early phase clinical data at the end of the presentation from these programs. So how do we do gene editing? So first I want to start with an introduction to the CRISPR-Cas9 system. You can think about this as a 2-part programmable system where you've got a guide RNA that identifies the genetic target. You can think about this as the software of the system. This is the element that's reprogrammable and can allow you to target different places within the genome. You then have the Cas protein. In our case, this is a Cas9 protein. This is essentially the hardware of the system, and when that associates with that guide RNA, that's responsible for targeted DNA editing and basically the enzymatic activity of that CRISPR-Cas9 combination. There's several key features I want to mention about this system. It is selective. It can be designed to be highly potent. It potentially addresses any therapeutic site, and you can also use this to target multiple DNA sites. So the focus of our 3 phase 3 clinical programs right now is in vivo delivery of gene editing. So this is where CRISPR is actually the therapy itself. We're delivering the CRISPR machinery and doing a gene edit inside the body. Our focus right now is on genetic diseases and treatment of genetic diseases through in vivo therapeutics. There's many strategic advantages that we think about by approaching therapeutics this way. You have that potential for single dose treatments that I've already mentioned. We use a systemic non-viral delivery of CRISPR-Cas9. We think this is important from a safety perspective. It offers some advantages in that there's transient expression of that CRISPR machinery. We want to deliver the CRISPR-Cas9, make the gene edit, and have that machinery disappear quickly from the body. Our lead programs are focused on gene knockout, but you'll also have the potential to do gain of function by selecting the appropriate editing tool. And then our lead programs are also focused on liver delivery right now. LNPs are well known to deliver to the liver, and I'll talk a little bit more about that mechanism, but we also envision and work on ways to deliver to tissues outside of the liver and look at potential for extrahepatic applications in the future. So a little bit more specifically on a liver LNP program or lipid nanoparticle program where we're doing in vivo knockouts and how this CRISPR mechanism of action works. So we start with a lipid nanoparticle drug product. This encapsulates a targeted gene-specific guide RNA as well as a Cas9 mRNA. This is delivered by an IV infusion to the patient. You get uptake in hepatocytes. This is a well-known mechanism where you get an ApoE binding to the surface of the LNP particle. You then have an LDL receptor-mediated endocytosis process that happens within the hepatocytes taking up these particles. You have endosomal escape, and the CRISPR machinery is then present in the cytoplasm. That Cas9 mRNA translates to Cas9 protein, associates with the guide RNA, forming that RNP complex that translocates the nucleus, and then can find a complementary genomic DNA targeted binding site. That creates a double-stranded break, and through endogenous DNA repair mechanisms, you can create an insertion or deletion at that particular site. So that's the direct action of the drug, and the intended effect that we're looking for here in our lead programs is reduction in a circulating protein or total plasma protein reduction of the particular target that we're knocking out. So to do this, we have to develop multiple drug substances as well as a drug product. So as I mentioned, we have that single guide RNA that is one drug substance, and that identifies that genetic target. The specificity of that guide RNA is created by the 20 nucleotides on the 5' end of that guide RNA. We also have a Cas9 mRNA drug substance, and these are co-formulated in a lipid nanoparticle drug product. This lipid nanoparticle is a multi-lipid component system, also containing a lipid that is a novel excipient. So I'm going to transition a little bit and talk a little bit more about how we design our guide RNAs from both an efficacy as well as a safety perspective. So there's several key attributes that are really important as we look to identify therapeutic guide RNAs. So if you think about this, you need to have high precision. We need to ensure that we're editing the genome at the intended target site or have the appropriate on-target edit that results in a particularly desired pharmacological outcome. In this case, we're knocking out a gene and reducing a circulating plasma protein. When we design these guide RNAs, it's also important that the target binding site is conserved across the patient population, so we can, with a single drug product, treat patients with this disease. And then certainly, last but not least, we're designing these guides and selecting them to ensure that they're highly potent as well. In addition to that high precision, we need to make sure that we have high accuracy. So we want to avoid any verified unintended edits elsewhere in the genome, often referred to as off-target edits. We also need to avoid DNA structural variants that may be associated with toxicity, and so that's part of our screening paradigm to look at these potential safety events. And then as part of that, we want to establish that safety window. So we want to make sure we have a wide window in terms of genotoxic safety versus expected therapeutic exposure, and I'll show you an example of that later. So our selection and qualification platform for guide RNAs starts from an informatics perspective, looking at the gene target, as well as then designing sequences, guide RNA sequences, and selecting and ranking them. These are then synthesized and formulated into LNPs, and we're progress those into in vitro screening, where we've got primary and secondary screens, where we're rank ordering and selecting highly potent guide RNAs at that point. Lead guide RNAs then go in further into that specificity screen, where we're looking at potential for off-target editing, and again, selecting highly potent but also highly accurate guides. And last, we do a structural variant analysis really on the top few lead candidates. So the overall goal of this is to ensure that we're selecting guide RNAs with the highest on-target activity, as well as no detectable off-target potential at multiples of our intended human therapeutic dose. So creating, again, that safety window through exposure. To give you an example of some lead guides, we put these through primary pharmacology assays. On the left, looking at in vitro data. This is done in primary human hepatocytes. Again, liver is our tissue of interest for these lead programs. What you can see in a dose-responsive fashion is an increase in gene editing. In this case, we're looking at the TTR locus in blue. As you increase gene editing, you therefore see a reduction in the TTR mRNA expression, as well as a reduction in that TTR protein. So again, we're getting and recapitulating that desired pharmacological effect in vitro. We can also take and use a humanized transgenic mouse and demonstrate primary pharmacology in vivo. On the graph on the top, we're showing a dose response in terms of editing at that TTR locus, and you can see that matches with a decrease in the reduction of serum TTR on the bottom graph. I just want to point out too that the primary human hepatocyte assays are useful in establishing that early pharmacology. We also use that as the basis for our drug product potency assays and understanding that from a release perspective further on for our drug product. There is a lot of work, though, that must go in from taking primary human hepatocytes into a potency assay that can be used in a GMP setting. But as you can understand, we're looking both at the genomic edit as well as reduction in the protein when we look at drug product potency. And I'll just talk briefly about our off-target workflow. As I mentioned, this is part of our guide RNA design and selection criteria. So this is a two-step workflow that we use. The first step is really designed to look at discovery of all potential off-targets. This is a combination of promiscuous assays. So the first is done through in silico or Cas-off finder prediction. This is coupled with a biochemical assay called site-seq. And so what you're taking and doing from these different assays is aggregating all potential off-target genomic loci. And again, these are designed to be permissive and allowing us to interrogate appropriately all potential off-target sites. We then take these into the second step of our workflow, which is a cell-based verification, looking at true off-target edits. We're using deep sequencing or next-generation sequencing really to look specifically at particular sites and interrogate those. From an in vivo perspective and development of our in vivo therapeutics, we do the final step of that verification using that LNP drug product formulation, as I mentioned, in therapeutically relevant cell lines, so primary human hepatocytes for our lead programs. And we can also use dose ranges and escalation and concentration to generate kind of these super therapeutic doses. And I'll give you an example of that data moving forward. So this is just an example of an on-target and off-target editing experiment in primary human hepatocytes. You see the curve in brown. You see that dose-responsive on-target edit at the intended loci. We also had several sequences that we were interrogating as potential off-target edits. And what you see is a clean off-target profile within the pharmacologic concentration of the guide RNA, which is highlighted at that EC90 or expected exposure that we would see in a patient. I highlight this tool as well because it becomes an important characterization tool as we progress in development. As we manufacture guide RNAs, we've done a lot of work that I'll talk about a little bit in terms of improving process and analytics, but in order to make process changes in the future, we do use this as a characterization tool to go back and check to make sure that we have not changed that off-target editing profile. So I'll transition and talk a little bit more about the CMC aspects of our programs and how we use manufacturing platforms as well as analytics. So we really want to take a platform approach to manufacturing for these multiple drug substances. So the first one is a single guide RNA. These typically for our lead programs are going to be about 100 nucleotides in length. Again, I mentioned that specificity of the guide RNA is those 20 nucleotides at the 5' end. And that's the element that we are changing in subsequent knockout programs to enable us to target different genomic loci. We use standard solid phase phosphoroamidite oligonucleotide synthesis. In contrast, Cas9 mRNA, our second drug substance, uses an enzymatic synthesis approach, much larger RNA, about 4,400 nucleotides in length, and this encodes for the SpyCas9 protein. We have multiple lipids within our formulation. This includes compendial, non-compendial, as well as a novel excipient. So you can envision that there's a variety of different regulatory requirements as we do the development of this formulation. And then we have the LNP drug product itself, where we combine both of our RNA drug substances in an LNP, and this is made by a self-assembly process with controlled dilution. So for each of these different manufacturing processes or platforms, we require about 15 to 20 analytical methods, really just for release of each of those components, as well as the drug product, with additional assays needed for characterization. We have multiple cell-based assays for both drug substance and drug product as well. So I think you can envision that there's a lot of work in developing both the manufacturing processes as well as the supporting analytics to bring a program like this into the clinic. So if we drill down a little bit more on guide RNA manufacturing, this is just a general schematic of unit operations that one uses. You've got an upstream process with solid phase synthesis, followed by cleavage and deprotection, and then a downstream purification process, really focused on removal of impurities and getting that pure product out of that. Guide RNA is typically synthesized in the 3' to 5' direction. And really, we're leveraging much of what's already known from an oligonucleotide manufacturing perspective. There's many marketed products with siRNAs and antisense oligos. They are much shorter in length, on the order of maybe 20 to 30 nucleotides. So there's definitely challenges that we have faced in developing both the manufacturing process as well as appropriate analytics to support development of a 100-mer process as well as characterization of that impurity profile. But in general, because we are leveraging much of what's known in oligonucleotide manufacturing, we've got similar raw materials, and really your approach to the process and the control strategy is the same and independent of oligonucleotide length. We do feel that once you have an established guide RNA process, this can be applied across those target sequence changes. So if we go into a little bit more details around the technical challenges of guide RNA manufacturing, you have to think about this upstream process being a nucleotide by nucleotide addition. So the theoretical purity of this crude product is really proportional to that per cycle efficiency or the efficiency in which you can add that nucleotide. And you see you have an exponential decay in full-length product purity with oligonucleotide length. So if we just focus on the green line, I'll go through an example here. If you think about a traditional 20-mer with a 99% per cycle efficiency, you can get roughly 82% purity in that crude product. In contrast, if you think about a 100-mer, just theoretically with the same per cycle efficiency, you would end up with about 37% purity in that crude product. So purification and development of that purification process becomes incredibly important as we look at longer oligonucleotides. It also increases the complexity of that impurity profile. Theoretically, you're going to be able to produce more distinct impurities from this process. So there's a lot of space and a lot of work that we've done in really optimizing, not only the synthesis process itself, but purification. And there's an inverse correlation, certainly, as you look to purify a full-length product, you are going to decrease your yield. And so that's a lot of development that has to be done to make these processes commercially viable moving forward. In general, there's been recommendations in the field for a full-length product purity of 80%. And so that's been a lot of work that we've done between early and late stage development, really to improve that full-length product purity as well as characterization of that impurity profile while really increasing or even maintaining process yield. Again, making sure we have a commercially viable process that we can move forward. Just a little bit more on analytical methods. While there are numbers out there of what full-length product purity should be, I think everybody in this room recognizes that's really truly dependent on the analytical method in which you use. If you look at an identical lot of material and you look at two different chromatography methods, we'll just call that method B on the right, and I see a purity of 88%, I think we're doing fantastic. I can look at method A, which has been optimized for the single guide RNA, and we realize that we have a much different full-length product purity. So really just emphasizing that, making sure that the analytical method is appropriate for what you're doing, but also development of orthogonal methods and abilities to characterize that impurity profile are really critical in the development of guide RNA processes. And I'll just highlight a little bit of some work that we've done on the purification itself. This is the purification process used in guide RNA. On the left, looking at temperature in that process. As a variable, we can increase that full-length product purity actually from 68.5% to roughly 80%. We can also increase yield in that process by playing with temperature. You can look at another variable being purification reagent, and you see that we can increase that purity from roughly 58 to 80% while roughly maintaining yield. And so it's been important as we've gone through development to make sure that we have a process that not only produces high purity, but also has improved yields coming out of guide RNA manufacturing. And as we've done a lot of these process changes, we've found, we have to do formal comparability assessments to be able to introduce these into the clinic. So we did a lot of work between early stage and late stage clinical development between process 1 and process 2. Our comparability included release data, extended characterization testing, looking at the guide RNA activity, as well as a detailed impurity analysis and a stability comparison. And so the chromatogram on the left is really just showing the difference between process 1 and process 2. You see an improvement in full-length product purity with process 2 and an overall reduction in impurity profile. And on the right, we were looking at a side-by-side after a thermal stress condition. And what we're doing is comparing the impurity profiles between process 1 and process 2. You see those are similar, indicating the degradation pathways are similar between these, and the manufacturing process itself and the chemistry that we're doing is not impacting that final guide RNA product. I mentioned off-target editing before, so while not part of our formal comparability assessment, it was an important element for us to go back and understand if there was any impact to that off-target editing profile. So we take guide RNA made with process 1 and process 2. In this case, we're looking at the KLKB1 locus, which is our intended on-target edit. Those guide RNAs are formulated into LNPs and then treated on primary human hepatocytes. It's also important to look across multiple donors. You see that on-target editing activity is maintained with the process change. And then if you look at the bottom lower half, we're really zooming in on MAPK1, which is a verified off-target at supersaturating concentrations. Just above the limit of detection here, you see no change in that off-target profile for this process change for guide RNA. So again, showing on and off-target consistency while we've been making this process change for guide RNA. So definitely significant time and resources are used in initial platform development. We want to be able to use that prior knowledge on the platform over the development life cycle, benefiting not only current, but also future programs. Hopefully this enables us to deploy subsequent programs more quickly with an increased probability of success. We've had a lot of regulatory learnings and interactions around the platform. This is a complex CMC package, and so the more that we do on educating regulators, the hopefully quicker and more expedient we can move programs into the clinic and get therapeutics to patients. And I'm going to end briefly on some highlights of some clinical data from NextZ, which is our investigational approach for transthyretin amyloidosis. ATTR amyloidosis is caused by an accumulation of misfolded TTR protein. This primarily affects the nerves in our heart. And the current standard of care in this space is chronic dosing for these patients. So the approach that we're taking here is to knock out that TTR gene with a single dose of a CRISPR-based treatment. This reduces both the wild type and mutant versions of that TTR protein, and we do aim to address two different manifestations of that disease, being polyneuropathy and cardiomyopathy. So key advantages, as I've already mentioned, potential for single-dose treatment, but really expecting lifelong stable TTR reduction, where we hope to halt and potentially reverse the disease with that deep and consistent TTR reduction. From a treatment experience, so this is meant to be a patient and provider-friendly experience, and I think it's important to highlight for in vivo gene editing and infusion of LNP. The day prior to administration, you're taking one orally administered pill. On the day of administration, you're taking 2 orally administered pills. You are going as an outpatient, basically sitting in a chair for a few hours, taking this drug product infusion, and then you're returning home that same day. And hopefully what you're gonna find is that we've delivered a CRISPR-Cas9-based therapeutic that leads to potential curative benefit for these patients. This is a snapshot of some of our phase 1 data with NextZ. This is after a single dose treatment. We're looking at reduction of that serum TTR protein. You see it is a deep and rapid reduction of that TTR protein. It is also durable in both populations, CM on the left and the PN patient population on the right, with residual absolute TTR concentration values roughly about 20 micrograms per milliliter, representing roughly a 90% reduction in that serum TTR from the baseline. So overall, this is showing the potential of NextZ to be a transformative therapy within TTR. If you take that a little bit further, we started to look at markers of disease progression. And again, this is early data looking at 12 months from our phase 1 study. We see that minimally we're stabilizing these markers of disease progression over 12 months for the patients. We're continuing to monitor this. We have an ongoing phase 3 looking at a much larger population to see if we really demonstrate clinical benefit in this space. I just want to mention too, the safety profile has been excellent. For Nex-Z, it's been well tolerated across all patients. I'm not going to go through the details in the table here, but we continue to progress this in clinical development. So I'll just highlight that the phase 1 results to date really show Nex-Z to be a potential best-in-class TTR-directed drug for the treatment of ATTR-CM. On the left, you see that Nex-Z serum TTR reduction. Again, it's deep, rapid, consistent, and durable. We're showing duration in this graph out to 12 months. The previous graph was out to 24 months. Again, this is ongoing, and we continue to monitor this. We've shown stability or improvement of disease markers. 66% of these patients had no worsening in any marker at 12 months. And overall, we have an encouraging safety and tolerability profile from this phase 1 study. And so we have 2 ongoing phase 3 studies. This is the Magnitude clinical trial for the treatment of ATTR-CM. This is a randomized double-blind placebo-controlled trial, a much larger N, approximately 1,200 patients that we're looking to dose in this phase 3. This has been ongoing for roughly the last year and will continue into next year. And this is a single IV infusion of 55 milligrams of Nex-Z. And then our second phase 3 study, much smaller in terms of the N, about 50 patients. This is a rare disease, being the hereditary form of this in terms of polyneuropathy. So again, a single dose infusion of Nex-Z, and we have both of these as active trials that we continue to enroll. So overall, I hope I've left you with the potential for CRISPR-Cas9 gene-edited therapeutics. And we really are committed to making best-in-class therapies a reality for people suffering from difficult-to-treat diseases. And with that, I will take any questions.
Hi, Kristy. I thought that was awesome, first of all.
Thank you.
You did an incredible job. My name is Steve Kennedy. I'm with CRISPR Therapeutics, so full disclosure. First of all, I'd like to thank Intellia as well for leading the charge into in vivo gene editing. It's really been impressive what you guys have done, and it really opened up the industry, so I appreciate that. And along those lines, I was wondering what your thoughts are on the next steps for in vivo editing, which in my mind are targeted LNPs, so that we can do something other than edit the liver. And then also like reverse transcriptase editors, which is going to, by definition, require a longer guide and kind of challenges what you just talked about in terms of guide synthesis.
Yeah, so I think kind of 2 separate questions there. Maybe I'll tackle the second one first. Certainly we look at developing different editing tools as well. There's a limit in terms of the number of knockout diseases that you can cover. Some of the writing technologies definitely require longer guide RNAs. I think, what we've shown over, several years of working in guide RNA manufacturing ourselves, we've been able to improve the process fairly significantly. I know some of these applications are going to require, 200 nucleotides, and there's options maybe for single synthesis, and, potentially you're looking at synthesis of 2 fragments and then bringing those together to enable kind of new ways and new editors. I think, your first question is a little bit more around delivery. The liver is fantastic for LNPs. We've shown that, thus far. I think LNPs still have a lot of future as we look at liver-directed delivery. Getting outside of the liver into extrahepatic delivery, I think we saw some examples earlier today of other types of delivery systems, whether or not that be LNPs, targeted LNPs, virus-like particles and polymeric nanoparticles came up, but I'm a delivery person at heart, and it is a very difficult challenge to get outside of the liver, especially through systemic administration. So hopefully a lot of us in the room continue to work on this and show progress in that space.
Any other questions?
Thanks for the great talk. I think you mentioned that you test the guide RNA initially in PHH and then moved to humanized mice. I'm curious, have you ever seen any inconsistency moving from in vitro to mice? And extend the question a little bit, moving from mice to non-human primate.
So the humanized mouse is really just used as a confirmation of primary pharmacology. So the guide RNAs, need that kind of human background. And so it's kind of a little bit of an outside of experiment of our normal screening paradigm. To address your question between mouse and NHP, certainly we've done a lot of work looking in mouse in terms of a formulation perspective as we develop this. I would urge others to really go into non-human primate because that is the bar when we talk about delivery systems for establishing whether or not your therapeutic is going to work, at least as it relates to LNP and liver-directed delivery.
Any other questions? I have one quick one. Sorry.
Okay, I won't leave.
So, we were just a tiny bit late, so I'm going to be cheeky and take us over a tiny bit. So you were explaining the challenges with chemically synthesizing oligos from sort of 200-ish. Is it still worth those challenges over, say, using a biological system to make those oligos?
So I think, one of the things I didn't mention too much about, but we require certain chemical modifications in a particular pattern for our guide RNAs. So when you go to biological systems, introduction of those chemical modifications at specific sites becomes very challenging.
I see.
So it really depends on the application and what kind of chemistry you need on that guide RNA.
Okay, that makes more sense. Thank you.
But great question, though.